Okay, so welcome back for lecture 3 of Unit 3. So first, some updates. First, by now you should have completed the Pocket Mouse lab as well as the Antibiotic Resistance lab. And last night, hopefully you submitted the Unit 2 Reflective Writing. And then Unit 3 Homework 1 is due by noon today. So if you're late on any of these, just get them to me. Being a little lenient with due dates at the moment for the time being, but we're going to try to get back to normal this coming week. So if you turned in the Pocket Mouse lab, I just gave you 100%. I will post the answers to that and go over them in the next lecture once everybody has that to me. And then on the antibiotic resistance lab, I curved that a little bit, gave everybody 15% back. So in terms of the homework assignments, make sure that you guys are referring to the lecture slides while you do them, all right? I want you guys to be getting points on these, and this is really to gauge your understanding of the material, so it's important that you are using your resources as you're doing them. These homework assignments, of course, are open note, and it's really to kind of help you learn. So just make sure that you're going through. I'm posting all the slides even without the videos on Moodle, so you can just pull those up and go through them as you do the homework. A couple other things. Remember to keep checking the Google Sheet for upcoming due dates for assignments. Additionally, I'm going to keep updating your grades on those individual Google Sheets that I sent to all of you, so you can check that anytime you want to see your grades for recent assignments. And then finally, I posted a feedback form for this new online instruction that we've been doing, so that's available on Moodle. It's at the top near the announcements and such, and you don't have to do it, it's optional, but I would definitely appreciate hearing your thoughts so far, whether whether you have suggestions for different tools that you'd like me to be using, things that you think could enhance your learning experience, or if everything's working for you, I definitely want to hear that too. So should only take a minute, so feel free to do that. Okay, so for today, continuing in Unit 3 on evolution. Today, mostly what I'm going to do is just kind of a review of the key concepts that we've covered so far. So just try to drive home some of these points about evolution, some of the take-home messages, and hopefully you're getting a good grasp on the forces of evolution and how evolution occurs over time. And then I'm just going to touch really briefly on the field of phylogenetics and what it's useful for. And then we'll dive into the Finch lab today, and that'll be due by 11 p.m. So get that done, hopefully just kind of use the normal class time for that and upload that to Moodle underneath the today's classes folder. Okay, so in terms of review for key concepts. So people to remember in terms of kind of building evolutionary theory. First, this guy Buffon, right, he had the idea, kind of the first to propose this idea that species could share a common ancestor. Then there was Lamarck, and while Lamarck has been, kind of discredited, he was still important. So Lamarck had this idea of the need-based acquisition of traits, right? So when you think of Lamarck, you can think of this idea of like a horse basically morphing into a giraffe as the vegetation that it needs to reach gets higher and higher. So kind of this idea of spontaneous generation and acquisition of traits based on need, which we know that mutation is random, right? And so mutations have to pre-exist a selective pressure. So Lamarck's ideas have basically been discredited, but they were still important in this evolutionary timeline. Then there's Wallace, who was a direct competitor with Darwin. He had the idea and published the idea that all species arise from pre-existing species. And he called these, closely related species allied species. And then, of course, there's Darwin, who is the most famous in the evolution field, who proposed this idea of evolution by natural selection, which has been validated by a number of studies, which, some of which we've talked about, and we'll talk about another today. Which is called the Galapagos finches, or in some cases people call them the Darwin finches. Okay, so in terms of evolution by natural selection, so natural selection is the principle by which each slight variation of a trait, if useful, is preserved. So the principles of natural selection, first of all, variation exists within populations of organisms. So this is the first staple of natural selection, right? So variation always exists within these populations. It exists before selective pressures act upon that variation. There's always variation, and that's due to mutation. We know that. So 2, there's a struggle for existence due to limited resources, and basically there are always going to be more individuals born than will survive. And this is just due to, species wanting to basically, due to these selective pressures, species always want to keep reproducing. And so they're going to produce more offspring than that will be able to survive due to the limitation of resources. 3, individuals with favorable adaptations due to this limitation are the most likely to survive and produce offspring. And this actually doesn't necessarily have to be just survival, but traits can also be selected for based on just general reproductive advantage. That's called Darwinian fitness. Then 4, these favorable adaptations will be inherited and passed on to subsequent generations. Then over long periods of time, successful variations will produce differences that result in the formation of new species. So again, this idea of Darwinian fitness is the ability to produce offspring over generations. And so we're talking about individuals with favorable adaptations that are more likely to survive and produce offspring. And actually, in some cases, your Darwinian fitness doesn't necessarily reflect a favorable adaptation, but it just reflects the fact that you did produce more offspring. So I've talked about Genghis Khan in that regard before, and how he had kind of a ridiculous number of offspring, not necessarily because of any specific evolutionary advantageous trait, but just because that's the way that it happened. And so it's important to remember that in some cases evolution can also be somewhat random. And so some things to remember about natural selection anytime you're thinking about this is that, again, mutations and variations are not occurring based on need or selective pressures. They occur before that. Mutations are random and natural, right? So there's always going to be mutations as individuals reproduce, basically. They're not arising because they need to. So in other words, if an adaptive mutation does not already exist within a population, in order to adapt to a selective pressure, then that population may not survive once a selective pressure is placed upon it. So you really need to have a variation that's basically advantageous in a certain scenario before that scenario arises, or else you might face extinction, if that makes sense. So in other words, mutations always come first, or they need to come first in order to survive a selective pressure. The selective pressure can then act upon those mutations in order to drive the evolution of a population. And so this is basically the order of events in each case. It's going to be mutation, then selective pressure to act upon that mutation, which leads to the evolution of a species. So natural selection is our first and strongest of the 4 forces of evolution that we've talked about. It's also the only one that's an adaptive evolutionary force. But it's important to remember that other non-adaptive forces exist and can also drive speciation and evolution as well. So evolution is not always adaptive. Evolution can occur somewhat randomly in populations, and we have evidence of this, and we'll talk about this later, but we have evidence in terms of, for example, the Amish population in eastern Pennsylvania and the prevalence of polydactyly or extra fingers there. So that's not an advantageous trait, and yet it's been it's somewhat evolved in that population due to these non-adaptive forces. Okay, so what are some of the non-adaptive forces of evolution? The first is mutation, which is basically just a change in an individual's DNA. So this can be a change in your DNA sequence, or it can be a change in like your chromosome structure, a chromosomal rearrangement, things like that. So it's a change to the DNA sequence. So as an evolutionary mechanism, it's important to remember that mutation by itself is generally pretty weak. Why is that? It's because mutation is actually usually disadvantageous. So mutations are generally not good for an individual. Why? That's because species are already pretty well adapted to their environments due to the process of natural selection, right? So most species and populations that you see actually have a lot of traits that they already have that are useful for their environments. And so without an environmental change, basically, mutation is usually going to do more harm than good. That's not always the case. Mutation by itself can be a good thing, but most often it's not. And so for those reasons, mutation by itself is a non-adaptive force of evolution. It's also a non-adaptive force of evolution because it's random, right? So mutation occurs naturally and it occurs randomly. So that makes it non-adaptive on its own. In order for mutations to be adaptive, you have to have natural selection to act on them. So these forces of evolution are, while we have them basically assigned into individual categories, they're non-exclusive, right? So all of these things are occurring in a population at the same time. Mutation, natural selection, again, genetic drift, and gene flow are all occurring simultaneously, but they're distinct forces. So genetic drift is the random fluctuation of gene frequencies from one generation to the next. So again, since genetic drift is just kind of random, this is another example of non-adaptive evolution. So genetic drift is only really a powerful evolutionary force in small populations. Genetic drift generally is a force that's going to reduce genetic diversity. And one example of this genetic drift is the founder effect. And so again, an example of this is polydactyly or extra digits in Amish populations. There are also a number of other examples of founder effect. If you Google founder effect, you can find a number of these. And so, but while we do consider the eastern population, the eastern Pennsylvania population of Amish to be a distinct population, it's important to remember that they're still the same species as us, right? And so speciation has not occurred. So basically, evolution can occur without speciation occurring. So while this population has evolved this specific trait of polydactyly, they can still interbreed with other humans, right? And so if we had something such as gene flow occur, then we could actually probably basically eliminate polydactyly over time. And so gene flow, as I just mentioned, this is the movement of genes and or alleles from one population to another within a species. So it's another force of non-adaptive evolution, and there are two components to gene flow. The first is migration, and the second is admixture. So generally different populations of a species are isolated, usually by geographical location. So in the case of the eastern Pennsylvania Amish population, they are not necessarily geographically isolated, but they are isolated by different means, basically, right? In terms of the breeding pool. So if that stopped being the case, basically, then we could have admixture, and we could reintroduce genetic diversity into that population, and probably then you'd see evolution act in the opposite way on that polydactyly trait, and that would probably go away. So again, these forces of evolution are all occurring simultaneously. And while natural selection is a very powerful force of evolution, it's not the only one. And so there are non-adaptive forces of evolution that occur within populations in order to drive the frequency of different traits. So some other key terms for forces of evolution. Of course, first, evolution is a change in allele frequencies in a population over time. And a population then defines the members of the same species that regularly mate with each other in such a way that the alleles are exchanged throughout the population. So, the eastern Pennsylvania Amish are an example of a population. And I'm using them as an example a lot, but it's kind of, a simple example to use. So it's important to note that different populations typically have differing allele frequencies, and they're often phenotypically different, right? So while they're within the same species, they can look somewhat different from one another. This is, you can also think of like people from different countries, people from like Eastern countries versus Western countries are another example of different populations that have, somewhat phenotypically different traits. And yet we can still, of course, interbreed. The phenotype then are the observable traits of an organism or the physical characteristics. Population genetics is the study of the genetic makeup of a particular population as a whole. and the gene pool are the alleles within a specific population. Okay, so when we talk about evolution and speciation, let's basically compare and contrast what these two words mean. So evolution, again, is a change in allele frequencies in a population over time. So evolution can occur within a population without necessarily creating a new species. So that's how we get distinct populations and distinct traits within different populations of an individual species. Now, speciation, on the other hand, is the formation of new and distinct species, which of course occurs through evolution. So as I mentioned, a population, right, can undergo evolution without speciation occurring. And then speciation, basically when we think about speciation, when we think about two distinct species, usually distinct species cannot breed as effectively with one another as they can within individuals within their own species. So a way to think about that is like while humans and chimpanzees are pretty closely related, we could never interbreed, right? So that's speciation often creates two different populations that cannot interbreed with each other. That's not always the case, but it's generally the case. And speciation usually occurs following geographical isolation. So this is really important to think about. So one of the things that can create enough separation between two populations in order to allow them to basically evolve into different species is like a geographical barrier of some sort. And so if you have two populations that are somewhat separate but still interbreed frequently, then you're typically not going to have speciation. They're not going to become two distinct species. So oftentimes geographical isolation can be important in order to drive enough difference between two populations such that they become distinct species. And so after geographical isolation, the four forces of evolution, of course, can drive the allelic changes within an isolated population such that over time, as these changes accumulate, the populations become different enough that new species emerge, and they're basically less likely to be able to interbreed with one another. Okay, so that's all I wanted to cover for basically the review portion of this lecture. So let's dive quickly into phylogenetics. So phylogenetics is the study of the evolutionary history and relationships among individuals or groups of organisms. And so phylogenetics seeks to ask questions like, where do species come from, right? How did they arise? And basically, which species is related to which species, and which two species are more closely related. And so this refers both to the genetics of two different species and as well as their physical traits, their phenotypes. Phylogenetics can also ask how far back the last, basically in history, was the last common ancestor, and what traits might they have had. And so one way we can use this is genetics, and another is that we can look at the fossil record. And sometimes we'll dig up specimens, right, that are clearly like a last common ancestor between two species or a link along that evolutionary trail. So phylogenetics, usually when we talk about phylogenetics, we'll see these sort of phylogenetic trees, right, that represent the links between species and basically how far back in evolutionary time we are linked to each other. So these are the three kingdoms of life: bacteria, archaea, and eukaryotes, right? And you can see, different basically representatives of each. So like we have animals, fungi, and plants within eukaryotes. Bacteria, different types of bacteria, gram-positive bacteria, for example. And as you can see, this evolutionary tree basically tells you how far back each of these things are connected to one another. So as you can see, animals, fungi, and plants all evolved basically from a common ancestor way, way back in evolutionary history. And then much, much, much farther back than that, of course, basically a single-celled organism split into these different domains of life. And so some single-celled organism ended up being all the different types of bacteria that we see, and much farther down the trail of evolutionary history, these single-celled organisms turned into multicelled organisms and then branched off into the different basically representatives of the eukaryotes that we see today, including animals, fungi, plants, etc. And so this is a way to trace evolutionary history back to shared ancestors. And so while this is kind of a really, broad view of this in terms of evolutionary history, we can also use this to study more closely related species. And so this is basically a representative of primate evolution. And so as you can see, we as humans are closely related to chimpanzees. We have a common ancestor not so far back in evolutionary history. And then farther back in evolutionary history, there was another common ancestor. And so basically, this common ancestor at this node is neither a human nor a chimpanzee, right? It's something in between. And farther back, there was another basically species of ape that was that split species-wise into this common ancestor and also gorillas. So farther back in the evolutionary tree, we're genetically related to gorillas. And you can trace this back, right, to before, basically before primates split into the old world primates, or the apes, and the new world primates, like some of these monkey species. And so that's the basics of phylogenetics and how that works. And so that will be somewhat relevant to the next thing that we're doing today, which is the Finch Lab, or the Finch Evolution Lab. So just a little intro to this lab and how you're going to complete it. Basically, the first thing you should do is watch this video. There's a YouTube link to it on the Moodle site, so go ahead and watch that before you start the lab. But just a little kind of summary of what that activity is and what's in the video. So basically, on the Galapagos Islands, there are these 13 species of finches. And in the video, the narrator is going to explain that basically there were these two researchers named Peter and Rosemary Grant who collected data on the beaks and some other traits of these finches. And basically what they found, and what other people have found, is that based on DNA evidence or phylogenetics, and so again, this is another example of a phylogenetic tree here, right? All of these species of finches are more closely related to one another genetically than they are to the mainland finch. And what this suggests is that actually one species of finch migrated into these islands originally, and from there, as the finches basically traveled between the islands and kind of became more geographically isolated from one another, different species evolved from there. And so these 13 species of finches all originally came from one common ancestor that came from the mainland. And so what the researchers were interested in understanding was basically how the speciation arose and how different selective pressures influenced the traits of the birds on the islands. And so the species that they focused on was the medium ground finch, which is found on the island called Daphne Major. And so they collected data on a number of traits about the birds, such as their beak size, their weights, and then they also recorded information about their mating and their offspring. And so this is some data that they collected on the beak depths, basically when they started this study. And so as you can see, the most common beak size when they started the study was around 8.8 millimeters, and this kind of follows, this Gaussian distribution of sizes around that, basically. So there's, there is this natural variation within this population that natural selection can act upon. And so they wanted to understand how that was going to occur over time, basically. So actually just 1 year later, the island underwent this pretty terrible drought. And so as you can see, a lot of the vegetation was dying, and they'll talk about this more in the video, but basically this drought lasted more than a year and killed off a lot of the vegetation. And what the island ended up with was these larger seeds. And so this is what the ground finches use as their food source. So when the drought hit, instead of the normal distribution of seeds that were basically available to them, the main food source that they had available were these large and hard seeds that you can see here on the left. And what resulted from this was that actually, because of the lack of resources, a lot of the finches died. Actually, more than 80% of the medium ground finches on this island died. And so this is that same graph again of the distribution of beak sizes, but this also has in black here on the bars the number of survivors from each group. And so basically what the researchers found is that the finches with the larger beak size, so as you can see, despite the fact that there are fewer finches overall with these bigger beaks, a lot more of them are surviving. And so what we call this in science is an observed versus expected ratio. And basically, if all other things were equal, then you'd expect, kind of like a proportional number of the representation of each beak size to survive. And so the black would actually follow the same trend as these red lines, but that's not what they found, right? They found that the birds with bigger beaks were much more likely to survive under these conditions. And so that's because the birds with the bigger beaks were better able to basically break these seeds. And so then when they studied the offspring of these finches, actually what they found is that even just in the very next generation, the average beak size had increased significantly, right? And so while prior the majority of the beaks had this average beak size of about 8.8 millimeters, in this next generation, the average beak size had shifted almost a full millimeter. And so this was a really significant change in that population, and really basically a very quick way to see natural selection, a population undergoing natural selection. Okay, so for our lab, basically what we're going to do is we're going to use data from this simulator that's based on these observations of the researchers, and basically based on the recordings of their data over time. So it's a bit unfortunate that this simulator no longer exists, but we have screenshots of the data that we can use in order to complete the lab report. So that's what we're going to do. And so what we're interested in understanding here is basically how precipitation affects beak size. So we can use these two fictional islands with the simulator. One's called Darwin Island, the other is called Wallace Island. Hopefully you can understand why by now. And then basically the input values, you can vary these different inputs to study different things based on what you're interested in knowing. And so what we're particularly interested in understanding are the how basically precipitation affects the average beak size of the population. So you can see that this is the range of inputs that you can use on the simulator. These are your options basically, and so these are the default values. So this is what we're going to start with for the first part of the lab report. So we're going to start with the input values of these 12 millimeter beaks and basically 20 centimeters of precipitation, which you can see on this scale is pretty low. So how does low precipitation affect beak size over time is kind of the question that we want to be asking. So when we input these values, we can go ahead and run the simulation, and basically this is what we see over time. So if we look at about 300 years from when we start the simulation with an average beak size of 12 centimeters, right? Over time, we see that the beak size actually increases due to this basically low precipitation and probably the availability of these larger seeds. So after looking at this data, which is going to be in the slides that says Finch Lab data on Moodle, so don't look at the data in here, make sure you're looking at the data within those slides, right? So then after looking at those data, you can look at your lab report and the questions in there. And so the first question is, how did the average beak size change on each island from 1997, basically between these 300 years? So going back to the data, we can see that the beak size on both islands increased, but it didn't follow exactly the same trend to get there, right? Nor does it end up at exactly the same size. So our answer for this question might be that the average beak size on each island increased over the initial 12 millimeters over the time studied. However, the increase was not exactly the same in each trial, so you're also going to, there's data from multiple trials in there, so you'll be examining that as well. And it was also not the same on each island. So that's an appropriate answer for number one. Another thing it's going to show you is how the population changes over time. based on your inputs. And so as you can see, the population on both islands also increases in this simulation. And so if we want to look at the specific numbers over time, we can also check that out in the field notes. So this is another part of the data that's going to be in the screenshots in that PowerPoint. And so as you can see over time, basically the beak size has increased over time. And so for your table 1 within the lab report, basically you would fill out the numbers based on these values, and make sure that you're not recording these exact numbers here, because this is not what's in your screenshots. But basically this is what you do, right? Is take these numbers from the final for each island and input it into table 1. So again, don't just copy these numbers because they're not accurate. But as you can see, this is what we get from the final beak size, right? In the year 2267 is this 10.42 for Darwin Island. And then for the population size on Darwin Island, we can see that the final there is 793. So that's what we'd record. And then for Wallis Island, alternatively, the beak size ended up being 18.5 and the population 666. Okay, so that's how you'll fill out the lab report, basically the table there. And so you'd expect that in the different trials between each island that the values will be pretty similar. And you're going to be comparing it basically you're not going to be comparing these values so much to each other, as you're going to be comparing the finals to the initial beak size. Okay, so based on these data and that first curve I showed you where the beak size increases over time, you might form a hypothesis that basically, and this is also part of your lab report, so you might hypothesize that a larger beak is more adaptive when hard seeds are more abundant due to low precipitation. And if that was your hypothesis, then you would predict that if larger beaks are more adaptive in areas that have low precipitation and thus harder seeds, then the beak size of the population will become larger over time. Okay, so that's basically how you run part 1 of this lab report. We're also going to look at, in part 2, we're going to look at the effect of the initial beak size on the final population size. And so here, we're going to input 2 different initial beak sizes on the different islands. So on Darwin Island, our initial beak size will be 12. And on Wallis, our initial beak size will be 29. And then we're going to look at how this affects the finch population over time on these 2 islands, considering that we have 20 centimeters of precipitation on both islands. And so as you can see, in this part, the data basically end up being that on Wallis Island, the population stays pretty steady over time, whereas on Darwin Island, the population is actually going to increase. And so this is another way to kind of test our hypothesis. And then there are also 2 parts for part 3, based on similar things. And so go ahead and go through and fill out this lab report, and again, get this done by the end of the day and submit that to the link on Moodle. And that's it for today's lecture. So again, make sure you're really drilling home these points on natural selection and evolution and kind of the mechanisms of evolution and the forces of evolution. And in the meantime, I'll be working on a key terms list for this unit that you can fill out, again, as your study guide. And then we will continue. We'll talk a little more about phylogenetics Thursday, and we'll go from there. All right, see you next time.